Yes, just going up.
So the same goes to oxygen. When we talk of urea, when we talk of CO2, hormones, they come from the endocrine glands. The destination is the target organs, and the fate is for coordination of transduction. And also we have mineral salts. Their destination is to the bones and the teeth, and they are for building up bones and teeth. And so this is basically some of the materials that are transported, their destination, what will happen to the end product, and their source. So in order to transport all these, whether in plants or in animals, we have to have a transport media. We have to have a media through which we are transporting our materials. So there are four major media of transportation, and they include the cytoplasm, the cell sap, or what we call latex, blood, and lymph. So to give you a hint of what I'm talking about, cytoplasm is found in all cells, all body cells. Cell sap is found mainly in plants like a leaf from a tree. There's something that oozes out. The juice that oozes out from the leaf is what I'm calling the cell sap. Blood is found in mammals, and so is the lymph. So the cytoplasm, it is found in unicellular organisms, and materials they are transported from one part of the cell to another through the cytoplasm. The cell sap, it is found in plants, just like I've told you. It is a concentrated solution.
Push it back, please. I'm here.
Okay, the cell sap is a concentrated solution in cell vacuoles, and it serves as a stronger solution. It is concentrated, highly concentrated in material. So it transports water and dissolved mineral salts from the soil through the roots to other plant parts. We also have blood. Blood, it is found in most animals, especially the vertebrates. So it is a liquid tissue, and it is capable of moving large materials over the entire body through blood vessels from the site of production to where they are required. We also have lymph, which is also another media of transport. So it is found in higher animals, the lymph, and it is similar in composition to tissue fluid, although it contains extra lymph. And there is no red blood cells that are present in the lymph. So it returns fluid to the main veins through opening in the subclavian, or what we call the left jugular vein below the neck, somewhere here, e.g. Lymph vessel of the luteal, which transport fatty acids and glycerol. I think the subclavian, this subclavian is a bone, is a type of bone. So the left jugular vein, it is somewhere here, the largest vein that transports these minerals. So these are the media of transportation that we have in living organisms, or rather in living things: cytoplasm, cell sap, blood, and lymph. So when we go deeper, we'll look at the composition, the functions, and the functions of the blood. So an average adult has about 5.5 liters of blood, or rather 5 to 6 liters of blood. The blood consists of liquid plasma, in which a mixture of blood cells floats. The plasma, it is a pale yellow fluid which is made up of 90% water, dissolved in it, including it includes also, or it constitutes also hormones, antibodies, proteins. I meant plasma proteins, gases like oxygen, CO2, enzymes, salts, digested food materials, and waste materials. So the major function of plasma is to transport the substances dissolved in it, as well as the blood cells. Blood cells include the white blood cells, red blood cells, the platelets, and it also participates in clotting of blood. Yes?
Yes, I'm here.
Okay. So as I've told you, there are two types of blood cells, or they're also called corpuscles. So blood cells, we have the red blood cells, the erythrocytes. We also have white blood cells, we have platelets. So there are about 5 million red blood cells in 1 cubic millimeter of blood. So erythrocytes are small, they're round and bicurvative or disc-like in shape. So a mature cell has no nucleus, and it has an elastic cell membrane which allows it to pass through narrow capillaries. That's why we have red blood cells transporting oxygen. They can pass through narrow capillaries. So hemoglobin, which is a component or which is a compound that is found in the red blood cells, the oxygen carrying pigment, I've told you hemoglobin is a component, it carries oxygen, and it is responsible for erythrocyte characteristic, the red color. So hemoglobin is what makes red blood cells red, rather. That's what I mean. So the red blood cells, they have a short lifespan of around 3 months or 1 day, and they become worn out and they are destroyed in the spleen. So worn out cells, they are ingested and they are taken out, and I've tried to explain to you. So worn out cells, they're ingested by the phagocytes, and hemoglobin is broken down to bilirubin and ferritin.
Okay. Yes. Hello?
So it is broken down to bilirubin and ferritin, and it is released into the blood. The liver stores ferritin and excretes the bilirubin via the bile. The bilirubin, it is excreted through the via the bile or through the bile, and it is what gives the fecal material its color. So bilirubin is the yellow color of the fecal material. So we have red blood cells. They are produced in the bone marrow and transport oxygen in form of oxyhemoglobin in small quantities and small quantities of CO2, and take part in blood clotting too. So we have white blood cells too. I told you of that, and also platelets. So white blood cells can also be called leukocytes, and there are about 7,000 leukocytes in 1 cubic millimeter of blood. They are large, they are colorless, they are irregular, and amoeboid in shape. So they are produced in the bone marrow, the lymph nodes, and the spleen. So the lifespan of leukocytes is about 13 to 20 days, after which they are destroyed by the lymphatic system. And there are 2 types of white blood cells. And So the two types of white blood cells, they include, do you know of any? Hello? 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 Can you hear me? So do you know of any type of the white blood cells? Any that you know?
Hello?
Sorry, I didn't.
Hello?
Yes.
Okay, now I can hear you.
Yes. So do you know of any white blood cells? So we have
White blood.
Yeah.
Uh,
Yes.
yeah,
the,
yes.
the types of
I'm listening.
Sorry. You know of any type? So we have phagocytes. They can be classified into phagocytes and immunocytes. Those are the two types of white blood cells. So phagocytes, they are found in the lymphatic system, where they ingest bacteria, viruses, and dead cells, and they help in preventing diseases. So they inject materials through a process called phagocytosis. Hence, these groups of white blood cells are called phagocytes. They can be subdivided into polymorphonuclear cells, because the name is because of their large nucleus, and monocytes, which have rounded nucleus. So those are the two classifications of the phagocytes. So
Mapits, could you send the note to me after this?
Yes, I will. I'll also send you some assessments, and so that I can see whether you've gotten what we are talking about. Is that all right?
Thank you.
Yes. So the polymorphonuclear cells, they are also called the granulocytes, and due to the presence of granules in their cytoplasm, the granulocytes are of three kinds. We have the neutrophils, acidophils, or what we call eosinophils, and basophils. So I've also talked of immunocytes, or what we call lymphocytes. So they are produced in the lymph glands, and they produce antibodies, which are proteins that bind to and destroy foreign materials. The antibodies, they bind to antigens or foreign materials, removing them. So some antibodies called agglutinins, they can cause clumping together of the microorganisms.
Recording wherever class. And it's blow softly.
A process
You're blowing too hard.
As they're shouting our hands, I'm not hearing the teacher.
As I've told you here, they produce antibodies which stick to the surface of germs and kill
Hello?
them. Yes?
Hello?
Yes, hello?
Okay. Yes.
Hello? Can you hear
Hello?
me? Yes, can you hear me?
Yes, I can.
Okay. So what I'm saying is that the lymphocytes, or rather what I've told you, the immunocytes, they can destroy microorganisms, foreign microorganisms, by production of antibodies that stick to the surface of the germs and they kill them. A way of removing them is through clumping together of the microorganisms in a process called agglutination. So the white blood cells, they defend the body against diseases, and they also produce antibodies which are immunobodies, which boost the immunities. So we also have others that are called antitoxins. So antitoxins, they neutralize poisons produced by microbes and other organisms. These are also types of lymphocytes. So the role of immunocytes in the
was
body
just cutting in the middle
against infection. So that is basically what we're talking about. So we also talk of which are also types of blood cells. Platelets are also called thrombocytes.
Yes.
They're tiny, they are irregular cells, cell fragments, which are non-nucleated. So
yes.
they are about
somebody wants to. It's on phone now.
So they are produced in large bone marrow cells. So platelets aid in blood clotting by producing the factor which initiates the blood clotting of the blood, so that when you are injured, there is clotting. So maybe to add on that, the lymph is colorless liquid. So these I've told you. This is maybe a summary of what I've talked about. When we talk of platelets, we can say that I've told you that they have no nucleus, but they have several organelles in them. So their function is to protect and to prevent blood loss through clotting after damage of the blood vessels. So they perform clotting by forming platelet plug in a wall of the damaged vessel, followed by production of substances called thromboplastin. The thromboplastin initiates blood clotting, or what we call coagulation. So hello? 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 So I've told you that platelets will initiate blood clotting, and the clotting or the coagulation of blood, it is a very important process because it prevents excess loss of blood and entry of disease-causing germs after injury of a blood vessel. So it is a complicated process which involves a sequence or a cascade of chemical reactions and several clotting factors. They have to be to play a key role there. So the first one, we have the release of thromboplastin. As I've told you, thromboplastin is released. So it is a coagulation factor or a clotting factor, which in presence of calcium, it works in presence of calcium ions, and it initiates a sequence of reaction that leads to the formation of a blood clot. So it can be released from two sources, extrinsic and intrinsic. So intrinsic thromboplastin, it is released by the platelets themselves, which clump together. We are together up to this point, which they clump together and they adhere to the wall of the damaged vessel, forming a temporary platelet plug in fibers or fibrin. So that brings about clotting, and you don't have excess loss of blood. So we also have extrinsic tromboplastin. When we talk of extrinsic, it is released by the damaged tissues. The damaged tissues release the thromboplastin. After it is released, we have the activation of prothrombin to thrombin. This is also another blood clotting factor. Hello? 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 Okay, so I will go on. So we have the activation of prothrombin to thrombin. So thrombin is a protein that is synthesized by the liver in the presence of vitamin K. It is converted to an enzyme called thrombin in the presence of thromboplastin and calcium ions. So initially you have to have the production of thromboplastin for the rest to be produced, or for the cascade to occur, for the process to occur. So we also have the conversion of fibrinogen to fibrin, which is the mesh that forms now the clot. So fibrinogen is a soluble protein found in plasma. So it is converted to insoluble fibrin threads by thrombin. So the fibrin threads form a meshwork which traps blood cells and plasma, forming a blood clot. So a stabilization of the clot, it requires the presence of coagulation factor, the fibrin stabilizing factor, and calcium ions. So that's how that's basically how blood clotting occurs. We are also going to look at the functions of the blood. So blood is essential because it transports oxygen using the red blood cells. So oxygenated blood is bright red, while the oxygenated blood is dark red. So I've tried to, here is an equation of what happens. We have hemoglobin combining with oxygen, forming oxyhemoglobin. So oxyhemoglobin is bright red, hemoglobin it is dark red, and we have now oxygen from the alveoli combining with hemoglobin. We also have the transportation of carbon dioxide. Carbon dioxide can be transported. Carbon dioxide can be transported by the blood in the form of carbonic acid.
Hello, ma. I'm so sorry. My internet tripped off.
Yes.
I'm so sorry. My internet tripped off.
Okay, that's all right. I actually, I thought that you left it while recording, so I'll go back. So I'll go back. What I was talking of is the function of blood. Uh, I've also talked of blood clotting. So blood clotting, it entails basically thromboplastin. Thromboplastin has to take part, thrombin, fibrin, and calcium ions. Those are essential for blood clotting. So it is a cascade, or it's a cascade of events that entails, first of all, the release of thromboplastin. The thromboplastin, it is a coagulation factor. Coagulation meaning it will bring them together, bringing them together, which in presence of calcium ions, it will initiate a sequence of reactions that will lead to blood clotting.
Oh,
We
no.
also have the activation of prothrombin to thrombin. So thrombin, thrombin is essential because it also leads to the conversion of fibrinogen to fibrin. Fibrinogen are soluble threads that are found in the plasma, but when they are converted to fibrin, they form insoluble threads that will form a meshwork, and this meshwork will trap blood cells and plasma, forming a blood clot, and that's how blood clotting occurs. So basically that's what platelets do. So uh, I've also talked of the transportation of oxygen in form of oxyhemoglobin. So oxygenated blood, it is bright red, while deoxygenated blood is dark red because it doesn't contain oxygen and only hemoglobin is available. So it can also transport carbon dioxide in the form of carbaminohemoglobin. So the CO2 is obtained from the cells, or as a waste product. It combines with hemoglobin. It forms carbaminohemoglobin. The carbaminohemoglobin goes to the lungs where the CO2 level, or rather the concentration, is low. It dissociates to form CO2 and hemoglobin, and CO2 is exhaled from the body, from the system. So that's how blood transports CO2. We also have the transportation of the excretory products like water and urea, and also transport of digested food. Digested food substances, they can be transported from the alveoli of the small intestines to cells, tissues within the body for the use and storage. We also have transportation of hormones. This is also another important function, and transportation of antibodies when they are produced. And also we have heat distribution. blood helps to distribute heat uniformly to all parts of the body and to regulate body temperature by getting rid of excess
Hello?
heat or sorry.
Hello?
Yes.
Hello?
Yes.
Hello?
Yes. Yes. I can hear you.
Okay.
We also have blood clotting. I told you that blood cells also have platelets. So the platelets, they initiate blood clotting when an injury is sustained at the blood vessels to prevent further blood loss and entry of pathogenic organisms at the point of injury. Um, so so this is a preview of what blood clotting is. I had explained earlier on. So this is what blood clotting, this is the cascade event of blood clotting. So platelets plug from to expose of damaged blood vessels to air and release a chemical which causes involuntary muscles in the damaged blood vessels walls to constrict, thus reducing blood flow. That is the first step. In a case, let's they reveal an enzyme called VATS inactive prothrombin into active thrombin. So the thrombin in the presence of calcium ions, it converts fibrinogen to fibrin, which forms a mesh of fine threads which traps blood cells by forming a clot that covers the wound and prevents further bleeding and entry of pathogenic germs. So it is a cascade of events. One has to be produced for the other to be activated. If you miss one, that's it. And in biology, for a cascade of events, you have to start from the starting point down up to where you get your results. So you have to, there's no shortcut of knowing this cascade. You have to know it. So the cloth forms a scab which drops off after a new skin tissue is formed in the injured area. So we also have the functions of the lymph. So the lymph, how I wanted to discuss on how the lymph first of all is formed, and uh, uh, and maybe look at what the lymph does. Uh, the function, as I've outlined there, it's a body defense and also absorption of fatty acids and glycerol. So in body defense, the lymph node produces the white blood cells, as I told you, also called the leukocytes. The disease-causing microorganisms. Sorry, I'm coming down with a cold, so I'm having trouble talking much. So, disease-causing microorganisms or the foreign material or foreign particles, they are and are filtered out in the lymph nodes. White blood cells, they are divided into two: immunocytes and lymphocytes, and phagocytes. Sorry, and phagocytes. Immunocytes can also be called lymphocytes. So that is one function of the lymph, and we also have absorption of fatty acids and glycerol. So the lymphatic system, e.g. the lutein, it is associated with the absorption of the fatty acids and the glycerol. So basically those are the two functions of the lymph. The lymphatic system, if we are to talk of maybe the lymphatic system, which entails now the lymph. So as I told you, lymph is a transparent, slightly yellowish fluid that is derived from intercellular fluids. So the letter, or rather, what I've talked of the intercellular fluid, it is found in the spaces between cells. So it is blood ultrafiltration. That is, capillaries on the arterial side, an ultrafiltration device. And that's how we get the intercellular components of blood, except the plasma proteins, which they have bigger size molecules and cells to pass through. Cells cannot pass through it, but blood cells, which are very tiny, they can pass through the blood capillaries. So this happens because of the high hydrostatic pressure. This happens because of the high hydrostatic pressure blood on the arterial side of the capillary bed. Remember, we said that when blood is transported, we have now we have the arteries, we have the veins, and then the capillaries, which are in the tissues. And in the tissues, they form the capillary bed. So that's what I'm talking about. So the pressure results from the pumping action of the heart. Blood coming from the heart through the arteries to the capillaries so that it can reach the tissues. So the pressure, it mainly results from the pumping of the heart and the high resistance offered by the small bore of the capillaries. So it forces fluid and low molecular weight components of blood out of the capillaries, forming now what we are calling the intercellular fluid. So the intercellular fluid, it circulates in the spaces between cells. It occupies the spaces between cells, and from it, the cells, they can take up oxygen and food materials and remove or shed off, not shedding off, or shed waste materials into it, into the intercellular fluid. So the intercellular fluid eventually returns to the bloodstream through two routes. Part of it returns to the circulation via the capillaries on the venous side by osmosis. Uh, this is the return through the venous side. It is facilitated, or it is enabled by the high osmotic pressure resulting from high concentration of plasma proteins and the fact that hydrostatic pressure on the venous side is very low. So when we talked of osmosis last time, we said that there are factors that affect osmosis, and we said that osmotic pressure is a factor that can affect osmosis and concentration gradient. So at this point, we are saying that we have high concentration of plasma proteins, and we also have the hydrostatic pressure and the high osmotic pressure. So that's why you have one root of it being osmosis, because of the concentration gradient. So, and then the rest of the fluid, it drains into the lymphatic vessels, forming what we call now the lymph. So the lymphatic vessels from various parts of the body, they come together to form one major lymphatic vessel, the thoracic duct, which drains into the subclavian vein near the neck. I talked of the jugular vein. So the flow of the lymph in the lymphatic vessels, it can be facilitated by the muscular movements around the vessels, compressing them pushing the lymph along. So that's how the lymph can move along. The compression and or rather the muscular movement, that's how they enable the movement of the lymph. And backflow, it is prevented by the presence of valves along the lymphatic vessel. We have valves just like in arteries and the rest. We can the flow of the lymph in the lymphatic vessels can alteration creates a negative pressure in the thoracic cavity. Inspiration is inhaling in of oxygen, which sucks. So when we inhale, we create a negative pressure in the thoracic cavity, which sucks lymph into the thoracic duct. So and expiration, it creates a positive thoracic pressure, which forces the lymph into the subclavian vein. So that's how the lymph flows. So all along the lymphatic vessels, we have small swellings, what we call the lymph nodes. So they are particularly numerous in the groin, where they are called the inguinal lymph nodes in the groin, around your pelvic and such. We also have them in the armpits, and they are called, in the armpits, they are called the axillary lymph nodes. And we also have them in the neck. So the lymph nodes, they are sites for the formation of the lymphocytes and the monocytes, which we say they defend against any diseases and any microorganism that comes in. So they are concerned with antibody formation. That's how they defend, and phagocytosis of the foreign materials. And in the lymph nodes, bacteria and other harmful materials are removed from the lymph. So that is how the lymph.
Hello? 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 I can't hear her again. Hello?
Hello?
Okay, now I can hear you.
Okay, sorry. I think there was a problem with my connection, sorry. So so that is how we form the lymph. We talked of the intercellular fluid. We've talked of ultrafiltration, which leads to formation of the intracellular fluid.
Yeah.
We've also talked of the lymphatic vessels, which form the thoracic duct. We've also talked of the lymphocytes, the monocytes, which will aid in the production of antibody that will defend against now the body. So basically, those are the functions of the lymph. And I've decided that at the end of every lesson, I'll be assigning you an assessment to see whether you've understood what we've been doing throughout the lesson. So you will answer the questions and you will send them to me. I hope that the moderator sent you the answers. I made a marking scheme of the previous work. Hello?
Hello?
Did the moderator send you the assessment, the biology homework?
Yes.
And you are comfortable with the marking scheme, or do you
I don't know whether what I read was correct or wrong.
Sorry?
I don't know whether what I wrote was correct or wrong. All I saw was just a bunch of red corrections, and I don't know if I'm wrong or correct.
Okay. So should we go through it?
Yes, that's fine.
Okay. For the remaining time, we can go through it because actually we started the class early. Uh, you can see from there. Hello. 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 Hello.
Hello?
Can you see from there?
I can't see the correction.
You can't see the correction.
I'm so sorry. What are we talking about? I thought we were going to review the last homework.
Sorry?
I thought we were going to review the last homework.
Yes, yes. And I'm sharing with you.
It's still on the functions of lymph.
Okay, so I'm sharing with you. That's what I'm doing right now.
Okay.
So can you see?
Yes, now I can.
Okay. So when we talk of diffusion, from what I've written, your answer was talking of only the movements. The definition of diffusion has to entail what is moving. First of all, we have now, you have to classify, you have to define what you are moving. So from your definition, I cannot see what is moving. You've only written the net flow or the movement of molecules, and it is across a semi-permeable membrane. So we don't know whether it's from high concentration or low concentration. So you have to include that. So where they are moving from and where they are going. Okay.
Yes.
I've also for the second question, I don't see a problem with it. Only that I added a few other examples of processes that are depending on diffusion. Also, how do the following factors affect the rate of diffusion? I have no problem with what you've explained. I think it's okay. For that part, I have no problem with that. But I have a problem with your definition. When you define osmosis, when you define active transport, when you define diffusion, you have to explain where they are coming from and where they are going. And from my definition, you can see that I've talked of the movements of molecules, and they are coming from a region of high concentration to a region of low concentration. And okay, so which I'm not seeing from your definition. So basically, that is that's the only problem. But the rest, I'm comfortable with them. So the roles of osmosis in plants. So it helps maintain the turgidity. Turgidity is not the shape. turgidity is how the cells, when cells have taken too much of water, they become turgid. That's what we're calling turgidity. So turgidity facilitates erection and the shape now of the plant cell. So I also added a few more so that maybe you can have a look and you can have more examples. So I don't have a problem with question 4. You explained quite well. I'm happy with that. So distinguish between arteries, veins, capillaries in terms of function and material composition. So I liked how you explained your answer, but you left something out. I've talked of in terms of function and material composition. You never talked of material composition. You only talked of function. So what they do, where they are, you've only talked of that, but I also talked of material composition. So from the table that I created for you, I found this easier for you to understand rather than writing a whole paragraph. So from the table, I have included the material. So relatively thick wall composed of smooth muscles and elastic fibers, and they also have narrow lumen. So this is what you are lacking. Also, the materials, you never talked of valves. Do they have valves? Do they? What happens? Because you have to include valves in them. That's a material that they have, and that's basically what you missed out on question 5. So when we go to question 6, discuss the distinguishing features of vascular bundles at every stage in their life. At every stage, I'm talking of when they are young and when they mature. I liked how you explained the functions, what happens, what goes on. The plant goes into secondary growth. They develop sclerenchyma, which help in support of wood and but die when they mature and give support to the tree, causing tree rings. So what we've talked about here is secondary growth. But what I want is the vascular bundles as they mature. What happens? We have first of all, as I've told you, as the cells mature, their cellulose walls are thickened by impregnation with tough substances called lignin. So this renders them permeable to water and solids. Unable to take up water and nutrients, the cells die, leaving a hollow tube, and that's how we have xylem vessels forming. So this is what I wanted from you. I have no problem with your explanation. It is quite good when we are talking of secondary growth in vascular bundles or in plants. But what I needed from the question is the features at every stage of development. When they are young, how are they? They have nucleus. When they grow, they lose the nucleus. The cells die and they are impregnated by lignin. They are lignified. That's what I wanted. So that's why I made the marking scheme. I expected that you would go through it and have questions. So do you have any other question?
No.
You've understood what the questions entailed?
Yes, now I understand. Thank you.
Okay. So for today's